Ever wondered how your genetic code actually comes to life? Today, we're diving into one of biology's most fascinating molecules, RNA, the translator, messenger, and sometimes editor of your genetic information. Stick around, because by the end of this video, you'll understand how RNA bridges the gap between DNA and life itself. Like and subscribe to receive more educational content like this. Let's start simple. RNA stands for ribonucleic acid, a close cousin of DNA. While DNA is your permanent library, RNA is like a temporary photocopy of a specific book you need for a job. Here's the key difference. DNA has two strands, the famous double helix. RNA is usually single-stranded. DNA uses the base thymine, T, but RNA swaps that for uracil, the U. RNA's sugar is ribose, not deoxyribose. It's got one extra oxygen atom. That tiny change makes RNA more reactive and less stable. Perfect for short-term work in the cell. RNA isn't just one thing. There are many kinds, each with a special job. mRNA, messenger RNA. Think of this as the delivery driver. It carries a copy of a gene from DNA to the ribosome, where proteins are built. tRNA, transfer RNA. The translator. It reads the mRNA code and brings the right amino acids during protein synthesis. rRNA, ribosomal RNA. The machine itself. It forms the core of the ribosome and helps link amino acids together. And there are even more. snRNA helps splice RNA. miRNA and siRNA regulate which genes get turned on or off. Together, they form a highly coordinated communication network inside every living cell. Now let's see how RNA is made, through a process called transcription. Initiation. The enzyme RNA polymerase binds to a promoter region on DNA, the start here signal. Elongation. The polymerase moves along the DNA, unwinding the strands and building an RNA chain by matching complementary bases. DNA A, RNA U, DNA T, RNA A, DNA C, RNA G, DNA G, RNA C. Termination. When RNA polymerase reaches a stop signal, it detaches, and the new pre-mRNA strand is released. This pre-mRNA isn't ready for action yet. It needs some editing. After transcription, eukaryotic RNA goes through processing before leaving the nucleus. Step 1. Splicing. Non-coding regions called introns are cut out, and coding regions called exons are stitched together. This is done by the spliceosome, a molecular editor made of proteins and snRNA. Step 2. Capping. A 5' cap is added to protect RNA and help it bind to the ribosome later. Step 3. Poly-A tail. At the other end, a long chain of adenine bases, the poly-A tail, is added. It protects RNA from enzymes that want to break it down too soon. And sometimes, RNA editing tweaks the actual bases, changing the meaning of the message itself. For example, in human mitochondria and some viruses, single bases are changed or inserted, creating different protein outcomes from the same gene. RNA editing gives cells flexibility. It allows organisms to fine-tune their proteins without changing the original DNA. That's like updating a recipe without rewriting the cookbook. In medicine, scientists are even using RNA editing technologies to fix genetic errors and design mRNA vaccines. The same principle used in COVID-19 vaccines that teach your body to make a harmless version of a viral protein. So, to sum up, RNA is the bridge between DNA and protein. Transcription copies the genetic message. Editing refines that message so it's ready for translation. RNA isn't just a passive messenger. It's a dynamic, adaptable, and sometimes creative molecule that makes life possible. If you found this helpful, hit that like button and subscribe for more biology breakdowns. And let me know in the comments. Should we cover translation next?